Hello, dear students. This is Dr. Shabana Begum, Associate Professor in the Department of Zoology, Maharani Science College for Women, Bangalore, presenting a lecture on the topic metamerism, that is part 2 under session 6. In fact, it is a subtopic under unit 1, that is animal architecture, pertaining to BSc Zoology first semester, paper 1. Students, in my previous lectures, I had spoken to you about the definition of metamerism and the two types called pseudo metamerism and true metamerism and its significance. Hoping that you have understood the lecture, let me talk to you about two more types of metamerism, that is the homonormous or complete metamerism and the heteronormous or incomplete metamerism. And the learning objectives are to study different types of metamerism, that is here homonormous and heteronormous metamerism, and to understand the evolution of metamerism in animals. The introduction to the topic. Once again, one can define metamerism or metameric segmentation, which is an architectural body plan in some animals in which the similar body parts or segments and organ systems are serially repeated one after another. The similar body segments are called the metameres or somites. Most of the scientists believe that the metameric segmentation appears independently at least 3 times during the period of evolution. These are the annelid and arthropod line, and in case of annelids, it evolved as an adaptation for burrowing. The chordate line, and in chordates, it evolved as an adaptation for free swimming movements or undulating movements. And also it evolved in the cestode line, that is in case of tapeworm. Now let us discuss about the homonormous metamerism. If the segments or somites of the animal are all alike, the segmentation is called homonormous metamerism, and it is seen in case of forms belonging to the phylum Annelida, such as the nereis, the earthworm, leeches, etc. Then each metamere here contains segmental blood vessels, nerves, nephridia, and coelomoducts. This is a primitive type of segmentation and is not found in any existing animal because a few anterior segments are specialized to form the head, which is called cephalization. A well-defined and well-organized head is lacking in annelids. However, the formation of a head is suggested in polychaetes by anteriorly placed structures and their association with parapodial cirri. The diagram in this particular slide is that of a nereis, which is a polychaete which exhibit homonormous metameric condition. What is heteronormous metamerism? So in case of arthropods and chordates, the segments of the body are dissimilar in different body regions and restricted only to certain organs. This type of metamerism is called heteronormous metamerism or incomplete metamerism. In complete metamerism, the segmentation is present on the entire body of the animal. So you can say it's homonormous. But in case of incomplete metamerism, the segmentation is not seen in all the body parts or organs. So hence it becomes heteronormous metamerism. The annelids show complete metamerism, whereas the arthropods and chordates show incomplete metamerism. The figure shows the schematic diagram of a polychaete, which show heteronormous metamerism. A new term, metametry. What is this metametry? So here the developmental period in which the embryo forms metameres is referred to as metametry. Based on development period or metametry, metamerism is of two types: homonormous metametry and heteronormous metametry. In case of homonormous metametry, it is a repetition once again. All the segments of an animal are identical and the segmentation is called homonormous segmentation. While in case of heteronormous metametry, the metameres have grouped together to perform similar tasks. In polychaetes, the segments between the head and the anal region are alike. So this becomes the example for homonormous metametry. Annelids you can always mention there. Then for heteronormous metametry, the highly differentiated segments are seen in many insects and crustaceans perform similar tasks. The examples for heteronormous metametry being the arthropods and the chordates. So this homonormous metametry is divided into two types: true metamerism and pseudo metamerism. It is again the repetition of the definition and types of homonormous metametry, which is discussed already under session 5 of metamerism part 1. Once again, if you want, I will just repeat. In case of true metamerism, the number of segments is constant for each species, while the new segments are added in front of the last segment called by GTM. Hence, posterior segments are younger ones. In case of pseudo metamerism, the number of segments is not constant, as new segments are constantly added throughout the life, and during growth new segments are added in front in the neck region, and hence the posterior most body segment is the oldest one. Simple elongation of the pre-existing segments results in growth in case of true metamerism, while in case of pseudo metamerism, addition of new segments from proliferation region in the neck results in the growth. In true metamerism, all the body segments are of the same age and at same stage of development. Proglottids, which are the segments of cestodes, they vary from one another in age and degree of development. In case of true metamerism, all the segments are integrated and interdependent functionally. They work in coordination and preserve the individuality of the body. And this individuality of the body helps in locomotion. Example for true metamerism is the form belonging to annelids. In case of pseudo metamerism, the proglottids are independent and self-contained units, as each of them have full set of sex organs, excretory and nervous systems. Each proglottid is productive unit developed for detachment. For example, such condition is seen in case of cestodes or tapeworms. So the outcome of the study, you can mention the metamerism can be defined once again as a linear series of segments of the body which are fundamentally similar in structure, but some of them perform special functions, hence they might not be alike in any single life form. Heteronomous segmentation, that is, the division of labor resulting in dissimilarity of the metameres of the different regions of the body, is found in the phyla Annelida, Arthropoda, and Chordata. Sorry, here the word phylum Annelida is wrongly typed. It should be only the Arthropoda and Chordata for heteronomous segmentation. The metamerism arises from teloblastic development, in which cells divide asymmetrically to form rings of small cells around the embryo. These cells proliferate into the many segments of worms and can be homonymous, meaning very similar, or they can be differentiated into a wide variety of functions. These segments are also called heteronomous metameric segments. With this, I will conclude my lecture on the topic metamerism part 2, that is session 6, before which I want you to answer few of the multiple choice questions, which are as follows. The first question is, when metamerism is not seen in all organs, the metamerism is called dash. The options are complete, incomplete, partial, and the none. That is, answer being option B, that is incomplete. Next question. Heteronomous metamerism is seen in cnidarians, ascaris, arthropods, all the above. So the answer being option C, that is arthropods. Third question. Homonymous metamerism is found in, options are polychaetes, cestodes, nematodes, and none of the above. So option is the A, that is polychaetes. Fourth question. Incomplete, that is incomplete metamerism means the segmentation is, options are not seen in all the body parts or organs, seen in all the body parts or organs, true in mollusks, and none of the above. So the option A, that is not seen in all the body parts or organs, is the correct answer, because here the incomplete metamerism means the segmentation which is not seen in all the body parts or organs. One more question. Besides Annelida and Arthropoda, the metamerism is exhibited by, options are cestodes, mollusca, chordata, and acanthocephala. So the option C, that is chordata, is the correct answer. Pseudo-metamerism is exhibited by nematodes, cestodes, cephalopods, and none of the above. So answer is the option B, that is cestodes. So the last slide here will give you some of the references or reference books and the website for you to go through it once again. Thank you.